Hi, so today we're going to talk about lipids and protein. So we're going to start off with lipids. So lipids, it's insoluble and the ratio of oxygen atoms to hydrogen atoms are lower than in carbohydrates. So there are three major groups. What are they? Before we move into the major groups, let's talk about its functions. So the first function is it stores energy in our adipose tissue, and it has a structural components in the cell membrane. And lastly, it's for some cases in the plants, it provides protection from water loss. So the three major groups are triglycerides, phospholipids, and steroids. Triglycerides are formed when one molecule of glycerol and combined with three molecules of fatty acid undergoes a process called condensation and triglycerides are formed. Let's move on to phospholipid. Phospholipid is formed during a condensation between one molecule of glycerol with two molecules of fatty acid and one molecule of phosphoric acid. Phospholipid molecule can be divided into two, which the first part is the hydrophilic head and the second part is the hydrophilic tail, which hydrophilic head is polar while hydrophobic tail is non-polar. Steroid. So steroid contains 4 rings of carbon and hydrogen atoms with various side chain. It is also soluble in organic solvent, but it is insoluble in water, such as other lipids. Let's move on to protein. So its monomer is amino acid, and this is the structure which has two groups, which is amino group and the carboxyl group. And below the C, where you can find the side chain R. So dipeptide. How dipeptide is formed is because it contains two amino acid, and which the amino group of amino acid join together with the carboxyl group of another amino acid, which they link between them. There is a peptide bond, and it is formed during the condensation reaction. And lastly, about protein, which is the 4-level structural of organization, which is the first one is primary structure, which is the sequence of amino acid by a polypeptide chain joined by a peptide bond, and the secondary structure, there is a chain that is coiled due to the hydrogen bonding, and two examples from it is alpha helix and beta pleated sheet. The tertiary structure is a 3D shape of polypeptide chain, which are maintained by hydrogen bonds and bonding formed due to interaction between the side chain R. So for examples of tertiary structure is enzyme. Lastly, quaternary structure is it consists of 2 or more polypeptide chains, and for example is hemoglobin. That's all for today. Thank you.